Today we're going to talk about the gene editing tool CRISPR and all of its benefits and drawbacks. This will be a very basic explanation of CRISPR, so for more information there are some links down in the description of this video to some sources that I used for this video. CRISPR is a tool that scientists and researchers use to modify genes and DNA. But to understand what CRISPR is and how it works, we first need to know what DNA is. DNA stands for deoxyribonucleic acid. It's an instruction set that tells your body how to make you. DNA looks sort of like this, and it's kind of important to this context because it is the thing that CRISPR is doing things to. Lots of people suffer from diseases that are caused by DNA being all messed up. Some of their DNA can be missing or different from normal, causing adverse side effects. Some common examples of this are sickle cell anemia and cancer. Many of these cases are unfortunately incurable. That is, if it weren't for CRISPR. The way we deal with these problems is actually really cool. The CRISPR enzyme goes into the cell and finds the DNA. It then searches along the DNA, looking for any problems in the DNA that are common with these diseases. So when it finds the problem, it takes the DNA and cuts it in that spot. At that point, it can either inject new DNA into the DNA sequence, or, and this is what it does most of the time, it just leaves it alone and the cell figures out what to do with it. The whole process is really cool. And what's crazy is that it's happening on this really tiny, unfathomably small scale. The technology required to do this is actually really new, so it's surprising that some CRISPR treatments are already in their clinical trials. Some of those treatments that are already in clinical trials include treatments for the two diseases I mentioned earlier, cancer and sickle cell anemia. Now, this isn't a perfect cure for every type of cancer, it's just one specific type of cancer that they're working on. And we probably won't have a perfect cure for every type of cancer anytime soon. That's just not realistic because every different type of cancer is vastly different in the way that we need to treat it. But the future is promising. In the future, we might be able to use CRISPR to upgrade our memory, upgrade our disease resistance, and increase the human lifespan. These things sound crazy now, but the possibilities are endless with this crazy new technology. But alas, nothing is perfect. And that's the same for CRISPR. There are some pretty big drawbacks that we need to consider as we advance in this technology. The main concern is ethics. Lots of people are worried about so-called designer babies. This concern is related to socioeconomic structure. Will the wealthy be the only ones who can afford these upgrades? It's something we need to think about and worry about as we advance. Another concern is consent. How can you consent to a genetic modification passed on by your parents? What are the ethics of that? The truth is, we don't really know yet. But unfortunately, those are the problems that we are going to have to face as this technology advances. But I'll leave you with this. Why should we stop this incredible advance in technology just because of some edge cases? What's the harm in trying to make advancements when we can always just go back and stop the research? In my opinion, the answer is obvious. Making progress is almost always good, even if we do find problems with it later. Doing something is better than doing nothing. And the good people researching CRISPR technology are doing one really good thing for the world.